Right. So we've got carbonic acid, which is being created by the combination of CO2 and water and the involved enzyme carbonic anhydrase. If you remember that enzyme, we've talked about it before, it's what helps to catalyze that conversion of CO2 and water to carbonic acid in either direction. So this is a bidirectional reaction. That can then dissociate. So that carbonic acid can then dissociate into hydrogen ions and bicarbonate. The bicarbonate is going to be exchanged across the basolateral surface for chloride. Oh no, I can't move my mouse. Oh gosh. Okay, there we go. So we're going to be able to exchange that chloride across the basolateral surface for bicarbonate. That is how chloride can get into the cell. So bicarbonate is leaving passively, right, indicated by the arrows. Chloride will come in, kind of hitching a ride actively. So that's going to be secondary active transport. And then that chloride will now have a gradient to move out of the apical surface according to that passive gradient. So that is how we get chloride into the lumen, as well as hydrogen ions into the lumen. Okay, and I apologize if I was all over the place there, but hopefully you can understand how those two things come together. So just to recap that one more time, we get chloride into the lumen because it is being exchanged for bicarb. It's coming into the cell, hitching a ride on the energy that bicarb is liberating as it moves out of the cell. And then it can move across the apical surface passively because of that gradient. It starts building up in here and it can passively move into the lumen. The bicarb is coming from the dissociation of carbonic acid. So carbonic acid being created by carbonic anhydrase by the input of carbon dioxide and water, and then that will dissociate to bicarb and hydrogen ions, and then the hydrogen ions can be exchanged across the apical surface as well for potassium coming in, but they're both moving actively. So they're both acquiring energy. This is primary active transport because both ions are moving against their gradient. And the energy for this is being used by hydrolyzing ATP. Okay, so that is how we get hydrochloric acid secretion from our parietal cells. All right. Let's talk about acid secretion here. So acid secretion happens under the regulation of the parasympathetic nervous system. Gastrin is also promoting acid release, and histamine as well. Now, because acid and pepsinogen are also secreted in sync with each other, pepsinogen from chief cells, acid from parietal cells, they both have the same stimuli, the same regulation for their secretion. So both are being influenced by the parasympathetic nervous system. Both are being influenced by the presence of gastrin, also coming from the stomach, from the G cells, and both are being stimulated from histamine. From histamine release. Okay, let's talk about how each of these phases are being regulated, or the secretions are being regulated from each of these phases. So the cephalic phase of regulation is going to be initiated by some of the things we mentioned earlier: the sight, the taste, the smell, the physical chewing, and then even the swallowing of food into initial parts of the esophagus. That is going to start stimulating acid release as well as pepsinogen release from the stomach as well. Okay, so in coordination with the parasympathetic input that's coming in, we also see that some of the cephalic stimuli also help with acid and pepsinogen secretion as well. Also, the parasympathetic nervous system stimulates gastrin release, and then gastrin also is responsible for acid and pepsinogen secretion also. Okay, so it's kind of this one loop, or one kind of domino effect, if you will. So the presence of food is stimulating the parasympathetic nervous system. Parasympathetic nervous system is going to stimulate gastrin. Gastrin is also going to secrete acid and pepsinogen release, but the parasympathetic nervous system can directly result in acid and pepsinogen release as well. Okay, just to look at that in this flow chart fashion here. So the sight, smell, taste of food stimulates the parasympathetic system. That is going to stimulate gastrin release from the G cells. And then both of these things will help to stimulate acid and pepsinogen release. So the parasympathetic system in a direct fashion, but then in a indirect fashion with the help of gastrin. Okay, the gastric phase, so moving down to the stomach here. The gastric phase is stimulated by the physical presence of food, so the presence of peptides and proteins, as well as amino acids, as well as the distension of the stomach. So the physical presence of food in the stomach is going to start the gastric phase of regulation. Remember, we said that we're starting the gastric phase in the stomach, the intestinal phase in the small intestine, whereas the cephalic phase is anything that's started in the head or mouth or oral cavity. This is going to involve both of our pathways. So this can be the short pathway if we bypass central nervous system. This will also include the long pathway if we include the central nervous system. But then we do have that option for either or. As far as the cephalic phase, it has to always be the long reflex. It has to go through central nervous system. And then this will help to directly result in gastrin release, as well as acid and pepsinogen release in a direct and indirect fashion as well. Another flow chart here, just kind of describing that. So the presence of proteins, the presence of peptides or amino acids, as well as the physical distension, stretching of the stomach by the contents there. That will send signals by the short reflex pathway and the long reflex pathway to the G cells, as well as the parasympathetic signal to the G cells as well. And then the G cells will directly stimulate the parietal and chief cells, but then our parasympathetic fibers can also stimulate the parietal and chief cells to release acid and pepsinogen. Okay, so a lot of feeding into each other, a lot of domino effects, one signal doing several different things, but then they all culminating to bring about the same result. Now we can see the inhibition of secretion in several different points. We can see exit or removal of food from the stomach can result in the gastric phase kind of being terminated or inhibited. We can see the increased acidity. So if there's too much acid being released in the stomach, that's another stimulus to kind of shut down acid secretion by inhibiting gastrin release. And then gastrin release will, the lack of gastrin will block that chief cell from secreting pepsinogen and the parietal cells from secreting acid. Okay. The intestinal phase can be inhibited if there's increased osmolarity, so too much fluid coming through, too much fat or acid present, or too much distension. So there's physically too much food present in the intestinal tract, and the duodenum specifically. That is going to be another stimulus to inhibit secretion and to kind of slow down, block that that movement of food coming through, or that phase of secretion. And then the long and short reflex pathways also do have a negative feedback as well. So we can inhibit acid and inhibit pepsinogen from being secreted if there's too much of that being secreted already. So kind of that negative feedback loop in terms of closing the cycle of both of our pathways, the long and the short pathway. Okay, I'll pause here and see what questions we have so far. So any questions that we have so far about secretions, about how they are promoted, as well as how they are regulated or inhibited. All right. So let's talk about some of the cells of the pancreas and the liver, and how they result in some of the secretions that we'll talk about. And we'll talk about how different hormones influence these types of cells. So first of all, we have the Astanar cells. These are the cells of the pancreas or the liver that are actually providing the basic components of the secretion. So they're providing a small volume of the primary secretion that will become pancreatic juice or bile. They're also providing a small amount of water, some electrolytes, some sodium, potassium, as well as the digestive enzymes that are present in that secretion as well. The ductal cells, so we talked about an acinus. An acinus is going to be the astanar cells themselves that are making the primary secretion. But then as the secretion moves along the ducts, the cells of the ducts are also modifying that secretion as well. So a large volume of fluid is being released into the duct. Bicarbonate is also being released into the duct, and all of that joins with the primary secretion that's coming from the astanar cells to result in the bile secretion or the pancreatic secretion coming from the liver or the pancreas, respectively. Okay, before I move on, a quick question here. How does the body decide between using the short or long reflex if there is no thought, taste, smell, or stimulation? So just to kind of clarify this here, great question. So essentially, if there is thought, if there is sight, if there is smell, if there is anything involving the head, it's automatically using the long reflex pathway, okay? So there's not a decision to be made. If there is anything involving the head, so the word cephalic means head. So any type of input that's coming in through our sensory perception, any input that's coming in through other areas of the brain, by default has to be communicated to the central nervous system, has to involve the long reflex pathway, okay? The short reflex pathway is the stimulation that can continue to happen by the presence of food in the stomach. So anything below the head. So the stomach being the gastric phase, the intestinal phase having to do with the small intestines. There's the continual regulation of secretion by the presence of food and chemicals in these other parts of the GI tract as well. These do not have to include the central nervous system, okay? So it's essentially that the short reflex pathway can happen in coordination with the long reflex pathway. That's one way to look at it. So essentially we can't have an exclusive short reflex pathway because unless there's some type of parenteral feeding, there is a way to put contents into the stomach without it going through the oral cavity. But that's kind of an exception. But essentially food usually goes through the mouth in order to get to the stomach. So the short and long reflex pathway can happen together. They can happen in coordination with each other. But the short is going to be what continues to happen from the stomach onward, where there's an opportunity or an option to bypass sending signals through the brain to the autonomic nervous system, and we can have a direct kind of a shortcut from what's happening in the stomach to the enteric nervous system, which is that local level of neuroregulation. Okay, that was an excellent question. So thank you for raising that to kind of really clarify how these two pathways operate. Okay. And any other questions, please feel free to drop them in the chat as well. They really kind of help me guide the conversation so I can, know what you are not understanding as well. Okay, so Astanar cells, we said, secrete the primary secretion, kind of the bulk of what's happening in terms of that secretion. Ductal cells kind of regulate the volume. So they're going to put lots of fluid in there. They're going to put the bicarbonate in there. That's also going to help neutralize the contents that are coming down from the acidic nature of the stomach. All right, let's talk about these two very important hormones here. So if you've understood nothing so far, make sure you understand the role of these hormones and what they're doing, as well as their stimulus. So what it is that stimulate the release of these two hormones. So CCK being cholecystokinin and secretin. So the enzyme and bicarbonate secretion are regulated independently by both of these hormones. And this is what helps the composition of the pancreatic juice, how that can change based upon the luminal contents. So an easy way to kind of express this is if there's lots of fats in the luminal contents, if you had a fat-rich meal, we're going to secrete more CCK. If You're also going to see more lipase in the pancreatic juice to help digest or break down those fats. If there are more proteins in your meal, you're going to see more proteases. If there are more carbs in your meal, you're going to see more amylases. So the contents of the pancreatic juice, as well as the hormones that are being secreted, are going to be determined by the luminal content. What is the makeup of the content that are in the duodenum? The hormones that are involved are CCK. CCK is going to stimulate the Astanar cells to secrete enzymes. That's their main role. They're also going to have a smaller role in stimulating bicarbonate release from the ductal cells. So the cells that are lining the duct are the ones that secrete bicarbonate, whereas the Astanar cells are the ones that secrete the enzymes themselves. So CCK, main role, the enzyme release from the Astanar cells. Secondary role, smaller role, is going to be the stimulation of bicarbonate from the ductal cells. Now, CCK also potentiates secretin release, okay? And because CCK also potentiates secretin release, and the function of secretin, which I'll talk about in a minute, is bicarbonate release, CCK is also going to be involved in this way in bicarbonate release as well. Secretin, on the other hand, kind of has that same relationship with CCK. Its main role is going to be ductal cells being stimulated to release bicarbonate. Its secondary role, or its kind of associated role, its smaller role, is to potentiate CCK, and we know that CCK stimulates enzyme release, and so they both have this symbiotic relationship. Secretin stimulates bicarbonate mainly, but then it also potentiates CCK, which stimulates enzyme release from the Astanar cells. CCK does the same thing. It stimulates Astanar cells to release enzymes, but then it also potentiates CCK, which allows for bicarbonate release. So at the end of the day, both of these hormones together result in enzyme release from the Astanar cells, as well as bicarbonate release from the ductal cells. Okay, I hope I didn't completely confuse you there, but they kind of have this symbiotic relationship where they potentiate each other, and then they potentiate the functions that each other are related or associated with. All right, let's talk about the stimuli for each of these hormones. So the stimulus for CCK is going to be fat and amino acids in the duodenum. The stimulus for secretin is going to be acidity in the duodenum. And that kind of makes sense, right? Because secretin is responsible for bicarbonate release from the ductal cells, its stimulus is going to be the acidity. If there's excess acidity, we want to have more secretin to help release more bicarb to help neutralize more of that acidity. All right. Same thing with the release for CCK as well. It's going to be stimulated upon fat present in the duodenum, upon amino acids present in the duodenum, and it's going to be resulting in enzyme release. So if there's more fat in the duodenum, more amino acids in the duodenum, we're going to have more CCK release, and more of that CCK is going to result in more of the enzymes being released, the enzymes being lipase to break down that fat, proteases to break down that protein. All right, so the stimuli are kind of lining up with the actions or the roles of these hormones as well. So just to kind of walk through this or illustrate this here, we have acid in the duodenum. That is triggering the chemoreceptors to stimulate the short and long reflexes. So again, short being just going to the enteric nervous system and stimulating the hormone release that we talked about. So from the endocrine cells, secretin being released. The long being that same route, but just now including the central nervous system, now including the autonomic nervous system, and then we get to the enteric nervous system. Okay, so both things can be happening simultaneously. We then have secretin release. Secretin is going to result in the bicarb secretion from the ductal cells, and then that can result in the neutralizing of the acid that's coming in and decreasing the acidity in the duodenum. Now, this is going to have a negative feedback loop. So once we tone down that acid, we then decrease the acidity in the duodenum, and we can start shutting off some of the release. The regulation of bile secretion into the duodenum, that's gonna be involving these two hormones as well. So secretin is gonna stimulate bile secretion from the liver as well. CCK stimulates the contraction of the gallbladder, as well as the relaxation of the sphincter of Oddi. So we talked about the sphincter of Oddi. It's present at the ampulla of Vater. This is the shared opening between the pancreatic duct into the duodenum, along with the common bile duct into the duodenum. And they're both being regulated by that sphincter, that strip of smooth muscle called the sphincter of Oddi. In order for bile to be released into the duodenum, we want the gallbladder to contract, to start vigorously contracting to expel that bile. We also want that sphincter to relax in order for that bile to flow right into the duodenum. All right. Secretin, on the other hand, is stimulating the actual secretion from the liver cells. All right. So in coordination, all three of these things result in bile being secreted into the duodenum. So secretin and CCK have several different roles, right? Just to kind of summarize these here before we wrap things up for today. So secretin stimulating bicarb release from the ductal cells, that is its main role. Also secondarily stimulating, potentiating CCK because, stimulating enzyme release because it is potentiating CCK. It is also stimulating bile release from the liver cells, and then its stimulus is going to be acidity in the duodenum. Okay, and then summarizing CCK here again, CCK resulting in its primary role, enzyme release from the Astanar cells, also potentiating secretin, and as a result, helping to have bile or bicarbonate release from the ductal cells, the stimuli for CCK being fat and amino acids, and then CCK also helping with contraction of the gallbladder, and then relaxation of the sphincter of Oddi. Okay. Now, these are really difficult to kind of keep together, so I'm going to encourage you to make a table, write out each of these hormones, and write everything they're involved in. All the roles of secretin, its primary role, its associated role, its stimulus, and then how it's involving bile secretion as well.